everyone, I'm Salma Quraishi. Welcome to Neuroscientists Talk Shop, the University of Texas San Antonio's neuroscience research podcast. Today is September 23rd, 2021, and we are back to an in-person format, which is really exciting. For those of you watching on our video cast, I will appear missing from the video feed because we're trying out some new stuff. But otherwise, I am here, and we're here with Doug Nitz, who is professor and chair of the Department of Cognitive Science at UCSD, where he also serves as the chair of scientific review at the Sanford Institute for Compassion and Empathy. Hi, Doug.
Hi.
So the Nitz lab works on the neural basis of spatial cognition, decision-making, and episodic memory. His research gets at how and where spatial knowledge is organized and used during navigation in specifically rodent models in your lab, I guess. The lab tests an array of spatial behaviors and examines the dynamics of neural activity taken from a broad network of areas that include hippocampal, CA1, subicular complex, as well as premotor and retrosplenial and posterior parietal cortex. So we have a neural basis of navigation expert group today with Isabel Muzzio. Hi, Isabel.
Hello.
And we've got Francesco Savelli. Hey, Francesco.
Hi.
And of course, Charlie Wilson, whose expertise is just transcendent and covers all things.
Hi.
So I was hoping we could just talk generally today about the how and why of computing spatial relationships in multiple reference frames at once, specifically with respect to the evidence for how representations for subspaces are built in neural space relative to a whole environment. So how we get from like the metrics of grid cells in this GPS system to building sort of larger understandings of the environment and the role of navigating path, how pathways and local sort of contributions sort of are built into global representation. So this relates specifically to, you've done a lot of work on the sort of local and global part of things, and it specifically reminds me of the retrosplenial cortex paper that you guys did in 2017. So could you sort of set up this idea of local and global for us in across this network as much as you can, and we'll get started there, I guess.
Yeah. Well, local and global, I'd say in the field kind of, in terms of the terms that used to be used, kind of mapped onto this idea of an allocentric space, which was almost always conceived of as the boundaries of the observable environment, versus egocentric space relative to the animal, which in most cases in the field meant the sequences of their actions as they move through space. And so naturally enough, those are two key kinds of frames of reference, the egocentric being the more local, you would say. But both a little bit limiting in the sense that allocentric space just means any external space that's not defined by the sensory organs of the animal, not defined, for example, by how something hits the retina. And then egocentric space should be egocentric spaces. So as the monkey literature will show you, across these different subregions of posterior parietal cortex, there's locations of targets relative to the hand, relative to the eyes, relative to the head and the eyes relative to the trunk. And there's lots of different spaces that kind of break down into allocentric and egocentric naturally enough. And then into local and then into local and global, I suppose. The global usually means the boundaries of the observable environment. But I think the important thing there is that sometimes those boundaries don't really exist as something you see. And sometimes I think we don't. We see wonderful things like grid cell patterns across a large environment, and we sometimes forget a little bit that the grid pattern that we see in our images and so forth it doesn't actually exist. But it's never there all at once in the brain. And so we sort of perceive it that way. And so I just think, at this point, a deeper level of thought about spaces and an opening up to all the different kinds of spaces that there are and what they mean is really interesting. So, good example of that is Isabel's work looking at the structure of environments. And a lot of work that's come out recently looking at distance and orientation relationships relative to objects, which in most of these experiments should probably be referred to as landmarks. They're rather large relative to the animal, and they're out in a space. So the field's kind of taking a turn in the right direction, I think. And so for me, I'll try not to hog all the conversation, but for me, it started in 2006 with this idea that the parietal cortex could map a location in a route, and that it wasn't dependent on the actions of the animal, and it wasn't dependent on the visual input to the animal, that it was really this sort of rather abstract concept of a shape that I'm moving through. And so, when I think most fantastically about the meaning of my work, I think it goes from a nitty-gritty neuroscience, but all the way up to the idea of, this is how a concept is built. There is this shape that I run through that I never see all at once, but I understand what it is. If I'm a human, I can draw it on a piece of paper and translate it. I build a concept. And so the spatial mapping is an opportunity to do that, right? To kind of go after concept formation. Yeah. You can tell I come from a cognitive science department now.
You mentioned things that are not there all the time. Like at any one moment, the brain has something. So one of the things I think about with a concept is that it's out of time. The concept exists without moment-to-moment changes. But if the animal is running on a path, and a neuron, we look at the data, and we can see, the neuron fired in this part of the path, and this part of the path, and that part of the path. But at any one moment, the animal was just in this part of the path, and only the neurons that fire at that part of the path are the only ones firing.
Right.
So, you triggered it when you said that all of that stuff that people see, like the grids and the grid cells, they're not all there at any one moment. And so I wonder, what is there all the time that we could make a concept out of that isn't constantly changing as we move around?
Right. What is there? That's where the computational neuroscientists need to come in to some extent. There's a study that was published a while back, a few years ago, where the animal goes through a semicircle path or through a full circle path. A full circle path being quite a bit longer. These neurons in the septal nucleus do not have spatial tuning, but the firing activity of those neurons relative to the ongoing 8 Hertz oscillation in the brain exhibits what we call phase precession. As it proceeds through this space, the firing of the neurons moves from late phases of this theta rhythm to early phases. And the thing for me is that it does that for the short path and for the long path. Once. It phase precesses once. So if the long path is twice as long, it doesn't phase precess twice, which means the rate of phase precession has changed. And that has to happen at the beginning, before you take off on the path. I desperately want to know what is there that can drive that change in the rate of phase precession, whatever that is the aspect of the system as a whole that
Yeah, I was thinking
that
something
respects
like,
the whole thing. I don't know if that's in a form of connectivity between neurons or probabilities of transitions between firing activity patterns. That's a real problem for neuroscience.
but that's what the thing you just gave is really great because it's a invariance. The concept is I'm halfway through this path or I'm all the way through the path, and it doesn't matter what the path is, halfway is still halfway. So that's kind of invariance is what creates the things that we normally think of as generalizations and concepts. So
Right.
it seems like that's really cool, and I thought there's something like that in your work where there are cells that fire, in the first half of the path and then don't fire in the last half, and another set of cells that have that in reverse, and then cells that are firing 4 times during the path
Right.
and presumably 8 times. And so you could create a sort of informational, by looking at all of them, you could locate the
You could always,
the path,
and
because in a sort of Fourier
yeah,
synthesis
exactly.
kind of way.
Yes.
And so that requires that those cells don't care how long the path is, just what one whole transit is, right? They have to somehow be normalized for the concept of that path.
The problem is, and I didn't mention this during my talk, is that that periodicity is context dependent. So if you have a neuron that fires at half intervals along one shaped pathway, it doesn't necessarily fire at half intervals on another one.
What would it do in the other one?
It might not have a periodicity. It might not have a periodicity, or it might have a periodicity of different frequency.
When you say a different context, are you talking the same shape but a different context or different shape?
Different, in my case, both, actually. So running the same shape through a different space in an environment, so moving the location of this pathway to a different space, but also running a different shape essentially through the same environment. Different shape through the same environment, but those two pathways share a subset of the locations that you move through in the room, and even that makes...
So those recordings were in the retrosplenial cortex, correct?
Mm-hmm.
And I was thinking about the studies done with human patients and virtual environments. I think Russell Epstein did these studies in which he saw activity in retrosplenial cortex only through paths. The virtual environment had locked doors and open doors. And when the subjects were thinking about the open doors, so there were possible pathways that they could transverse, then they saw activity in the retrosplenial cortex. So I wonder whether that activity is a mapping of the transversable space that the animal can afford, to move on.
Right, the affordances.
The affordances of the space. Is there any evidence, and have you put blocks in that? When you see these, the sequences of activity, have you tested putting blocks in those environments like you did in the original experiment to see if that activity is altered?
I haven't. I haven't done that. I was kind of hoping to do that in a different way.
Oh.
I was kind of hoping to take the circular track, which is useful because it doesn't really have a repeating structure except the full cycle, right? Or infinitely if you want to take any single period from another. But to impose hurdles, just little hurdles, it doesn't have to be much, just enough that the animal has to change its gait, has to know
Exactly.
where they are, and to see if I could impose impose a periodicity, to try to to try to get at whether the periodicity has something to do with the literal structure that they move through, or has something to do with their actions. And so, yeah, no, that's a really interesting problem. Certainly, I follow Russell Epstein's work pretty closely.
I wanted to ask you a question because during your talk at the very end, you showed some slides that you didn't have time to present, but it seemed to me
Because everybody was talking.
Yes, I know. We were so excited to have you in person for the first time that everybody was asking questions, me included. So I apologize for that. But
I apologize. That was fun.
people have, the Hasselmo lab and your student have shown these egocentric boundary cells that fire in relation to the borders and the direction of the animal. And I thought that you were showing that you have found object egocentric firing as well in one of the slides. I don't know if
It was in posterior parietal cortex.
It was in posterior parietal cortex, I think. One of the last ones, or not? In relation, did you have something that the cells were firing in an egocentric manner relative to the objects that were in the environment?
This was the pursuit, the target pursuit.
No. Well, if you want to consider the light that's flashed down on the platform that the animal pursues to be an object of some sort, and maybe that's reasonable, then yes, posterior parietal cortex does map that out pretty nicely. Explicitly, if I'm remembering right from what Andy Alexander told me about his egocentric boundary cell work, the egocentric boundary cells differentiate between boundaries and objects. And this was also true for the data from the Moser laboratory, the mouse medial entorhinal cortex object vector cells. It was in their work, a completely separate population of cells from border or boundary cells that have been talked about from the same laboratory and others like the Lever laboratory. They're kind of separate entities. So yeah, we'll see where that goes. I don't know. I am doing experiments right now where objects are moved around in an environment, so I'll be able to tell you a year or two from now whether the posterior parietal cortical cells really respond to objects or landmarks in an environment. But yeah, the location of the pursuit target, this light that's moving around on the floor relative to the animal, was definitely built into the parietal activity, and some hints at that were already actually out there in the literature from Rebecca Burwell's laboratory, where her environment visually is often characterized by these, I forget how the system works, but it creates images that can show up on the floor. The environment can be moved from time period to time period. And so she's seeing some responses like that as well. Yeah.
And a follow-up of this question is that we always, whoever is working on navigation, we focus a lot on excitatory cells and, especially for the spatial properties. But there is a paper by the Mosers that suggests that at least some subpopulations of GABAergic cells seem to code information about boundaries. I don't recall now if it's the parvalbumin cells or the somatostatic, because they differentiate between the two. So in your recordings in other brain regions, have you found that cells other than the excitatory cells code some aspects of boundaries or the environment or egocentric cell, boundary vector cells or something?
So yes. So in the cortex, you're recording kind of blindly. So what's an excitatory cell and what's an inhibitory cell, at least in my recordings, is not really known to me. I mean, that's going to change soon with all the powerful new tools that are out there. But the very fast-firing neurons separate themselves as a category of neurons. And in parietal cortex, they tend to have very similar firing patterns as to the excitatory cells, just a lot faster. But in the hippocampus, in the CA1 region, and in the subiculum, and in the dentate gyrus, we had a 2007 paper where we showed that hippocampal interneurons have place fields. And
Lauren Frank also has a paper showing that they have a spatial tuning, so it's quite interesting because in the field it's not so discussed.
It comes and it goes. Interneurons and their spatial tuning seems to come and go every 10 years or so. I went to the meeting with that data, and I thought, I did this cool thing, and then Bill Skaggs came up to me and said, that's really cool, but there's 5 other people presenting interneurons with place fields. It's just this horrible coincidence. But suffice to say, for interneurons in the hippocampus, there are what you'd call positive place fields, where the rate increases reliably over a space that, when you average it out across neurons, looks like the size of a place cell's excitatory field from the same dorsal ventral level of hippocampus. And then, because most of these neurons are firing on average at 20 hertz or so, you can actually see sort of kind of reverse or inverse place fields, where their activity drops to 0 in a particular location reliably, it's like a reverse. It's like a reverse place field. It has the same size as the CA1 excitatory cell.
As expected, if it was participating by disinhibition and helping the pyramidal cells.
Right. So I think for a place cell, it's as much about its excitatory inputs as lacking an inhibitory input at the same time. So that the aspect where the fields are the same size and more or less the same shape, that really impressed me. And we built that into a model of hippocampus that I did with a guy named Jeff Kritchmar, who's at UC Irvine now, but we were together at the Neurosciences Institute back then, and we built that kind of sculpting according to inhibitory cells into the model, and it really made a huge difference in our hippocampal model. Stabilized it and just made the spatial tuning much more precise. So
today you were talking about, when you started your talk, you brought up the idea of the cognitive map, and originally when people, with O'Keefe and Nadel, people were thinking about the hippocampus, but now we know, and you mentioned this today, that the cognitive map involves many brain regions. So would you like to discuss if you think that different brain regions are contributing different aspects? How is this cognitive map built in the brain, and what is the contribution, at least, of the regions that you are studying?
That's not too hard, is it?
Yeah, easy.
It's a very broad question.
A sub-question to
you
that.
can take the question if you want.
It's like, no, I'm not going to answer that question. But just to kind of add something to that question. So, O'Keefe and Nadel, in that book, they, if I remember correctly the terminology, they distinguish between what they call locale. I don't even know actually how it's pronounced, but it's written with a local, with an E at the end, locale, and taxon. So one is the idea of the cognitive map, kind of the way Tolman talked about it. So this allocentric representation of space, which is, what we talked about, and place cells. So the idea of place cell, going back to the ideal concept, right? What is invariant? We're to a first approximation, path equivalence, right? So, but then of course there is a lot of modulation in place cells about path, which is your study works. But the first idea of the phenomenon is that place cells can be modulated by a lot of variables. But the idea is like when the rat goes back to the same place, the cell fires again, whatever circuitous, tortuous path has happened. So that's the beauty of it, is like how does the brain do it? And then, so this idea, okay, you have an allocentric map that allows you, for example, to take shortcuts through unexplored environment, which was the idea of Tom. Then they say, okay, there is this Taxon system, which is more about like, navigating mazes and paths. And if I remember correctly, they say, okay, this is a different system, probably taken care of by the different part of the brain. And that's why I was like, I'm such a big fan of, your work and, that 2006 paper, because it was, so nobody knows what is this other system. I don't know that we know much about what this other system would be. And we know little, but to me, it was always like, well, is what Doug is doing, is it after the taxon? So like these paths, these progress, these cells that you found in posterior parietal cortex, that are neither egocentric nor allocentric, but they are, you call them in your papers route centric, meaning like, they just track this progress of the path. Is that the taxon? Did you find the taxon?
I often think of the taxon as a
well, that's what
a distance from a landmark that you're trying to approach, but there's no reason why that line can't be bent and be considered a route.
Routes.
At that
so like, if there's a
but I wouldn't say it's a taxon in the sense of being egocentric, where you solve the problem of getting to something by just approaching and keeping it in a certain egocentric relationship to you. I'm moving toward this landmark. I don't need to know where I am in the world.
Okay.
Right? So route centric, I would consider to be allocentric in this sense, which just means other centric, not egocentric.
Just not with respect to a XY Cartesian reference frame, but the progress along a particular route.
The progress along a particular route. So I wouldn't.
But anyways,
I wouldn't say I discovered a new.
Well, I'm asking. So the point is, okay,
so maybe not to say that, but just more to emphasize a different kind of space.
Yeah.
One that doesn't relate to boundaries.
Yeah, so that's exactly the point. So maybe let's put away the terminology and everything. But so yeah, this is a different system. Very different. It's not the cognitive map the way that it was originally intended. I mean, it's a cognitive map in the sense, okay, in cognitive science, for example, where cognitive map was used. I think about for example, Benjamin Kuipers, who actually I work with. There was any kind of spatial information that you have in your mind to actually navigate even mazes and routes, actually. Mm-hmm. But in the tradition of place cell and O'Keefe and Adele, I always thought, okay, cognitive map is something different from this system that you're investigating. And so the question is like, okay, how maybe the two things come together? Because, again, we go back to this idea of place cells and grid cells being GPS, right? I always say, what's not GPS? It's like, it fragments when you have boundaries. It's not really a perfect allocentric map that encompasses all the systems. So it's like, are these two systems coming together in some way, or you study a lot the commonalities of hippocampal cells and cortical cells, and
I think they're part of the same thing.
Okay.
I think they're part of the same thing, really. I think that what happens is we put the animals in these environments that are open and fully surrounded by a boundary, and then that becomes the space that we can find activity in. That becomes the space that's relevant to the animal when they're in there. And so we see the map in that form, but we kind of fool ourselves a little bit into thinking it's this two-dimensional map that is metric, right? There was just this paper published in, was it TINS, John Ruckelman, Elizabeth Buffalo, Lisa Giacomo, that's saying even this universal map, the grid cell map in the entorhinal cortex, is really topological. And there's a lot of other things that are consistent with that, not the least of which is when the animal's running on pathways through an environment, the grid map is no longer a grid map.
Right.
It's gone. It doesn't.
Well, but the issue is
So
there is a system there that somehow is able to parse space, however, not in a perfect way, but is able to kind of break up space in regular distances. And these regular distances have no relationship whatsoever with external landmarks, because even if they did, there are so many grids,
Right.
that the brain is producing. Of course, it's the grids that we see after we make the rate map and anything, but, they are different cells. So this is somehow internally calculated.
yes. So you have a system that's metric in the sense that you can update yourself in this grid cell network according to self-motion, and it seems somewhat, at least, metric in that way in the short term.
Right.
But
Right.
I think the question is, what's that for? If you say, what information do you get out of the entorhinal cortex in a
Right.
grid cell network? I think the most reasonable thing is to say that the only thing that really matters and that could really influence the rest of the brain over some long period or over some period of time is a relatively short period, maybe a few seconds. I mean, that's what I mean when I say the grid pattern firing over the entire environment. That doesn't really exist in any way in the brain, because only little snippets of that are transmitted to other brain regions
Well,
moment to moment. So
there are population of grids. So some models, for example, they postulate, I don't know if they're right, but they postulate actually there is a grid in the neural system with bumps of activity that are equally spaced, and it moves in lockstep with the animal. So if you're recording from one neuron, that also looks like a grid when you're on a rate map, because of the way the structure is imparted on that particular neuron. So I don't know, that's just one model. Like all the other models have more or less implicitly kind of produce that phenomenon. So, so a lot of cells at the same time, they might, well, at least probably they signal position. Then the issue is like how the brain might use, if at all, these regular distances that they get expressed at some point in the grid system. But that's a big question about it. I don't think anybody.
I think
lots of ideas,
that's
but
the
nobody
most
knows.
interesting.
Yeah.
I think that's the most interesting
Right.
question, though, because to the extent that it's a universal map, it's really not a very good map at all.
It's like you're trying to get around.
It's like I got a map I'm trying to use the same map for 5 different environments. It's not very.
It's not going to work.
So I think that like the shorter term, I've gone out this far, and then over this way, this distance. I think that's what we get out of that. And so I
it's
wouldn't,
just like why and how and yeah.
right, which is maybe explains why the light gets with Brandon, they through the lesion of the medial entorhinal cortex, and in the CA1 region, there's still place-specific activity. What's gone is the phase precession.
Yeah, but
What's gone is
usually
the
those
phase
experiments.
precession. It's sort of like the more short-term, how much space have I moved?
Right.
How much space have I moved through? I think that's the magic of the grid cell network.
Yeah. So in large environments, lesions of MEC brings significant disruption, or pharmacological inactivation, significant disruption on place field formation of place cells. So it's just, there is also these things about novelty versus familiarity. It seems like when you really look at the nitty-gritty details, somehow the place cell system relies on grid cells much more during novelty than during, familiar conditions. If by relying, you mean like, making a nice place field, which, we like nice place fields because we like to look, but who knows whether the brain even cares about it. But, as long as we, consider a normal place cell system like making nice place fields in a large novel environment, if you disrupt the entorhinal cortex, you do have significant disruption of places. So there's also that. It's like, why is it that the grid cell system seems to be more necessary in a large novel environment? So maybe because in a large novel environment, you cannot rely on landmarks because you haven't mapped the landmarks
You don't know them yet.
So you need some path integration in order to put the landmarks on your whatever internal representation. So I don't know, that's always been my pet theory, but I don't know if that's
I was trying
right.
to see the relationship between what you're saying and Isabel's original question, and I get it. So I think I just get it anyway. So
That's good.
when we ask people who work on navigation or on the concepts related to space and how the brain works on them, they always give you an answer that is some little fragment. And you say, but I've got place cells. You say, yeah, but how does that get you the concept of space?
Yeah.
They go, well, it's definitely going to be part of it. It's going to help. And you go, yeah, I think that's right. I think that would help.
No, it just follows that you do have a concept. It follows that you have a concept of space. It doesn't mean that you got your finger on that.
So then you ask somebody else, you ask somebody who works on grid cells, and they'll start telling you how grid cells do some contribution. But you ask, is that the thing? And they, well, no, but it is, but it's part of it. Pretty sure that's part of it. You go, yeah. And then
you
it
look
turns
at
out,
the posterior parietal
that's
cortex.
right. And so there are a bunch of things that have been identified that I think could possibly be said to be part of the solution. Maybe you're more hesitant about going.
No, I'm not
ahead.
hesitant. I think they're all relevant. I feel like sometimes we're trying to jam spatial cognition for your environment into one fixed idea. And I think if we just think a little bit about our own experience, I think that's not really true at all. I think that sometimes when we're conceiving our space, we're not really thinking necessarily about space at all. We might just be thinking about directional relationships. This highway goes off in this direction, and this one goes off in this direction. And yes, there's a spatial component that's implied by the angle between them, but it's kind of a murky idea, isn't it? And the only thing that you need to know is I need to go that way, or I need to turn from going this direction to going that direction, right? I don't necessarily need to know where I am in some bounded space. I might just need to know, oh, that landmark's there and that landmark's there, so my target is there. And so you're solving the problem different ways at all times. I think all these things are going on in the brain, and we keep trying to jam it into one system that operates in some rigid way all the time. And I think it's
that's our job.
naturally that the brain is much more human.
We have some
than that.
set of observations, and we're hoping that they explain everything. And so we always try to phrase it so it sounds like they do.
We get a little too computational sometimes. Right. But as Gerald Edelman reminded me several times, the brain is not an equation. So
It's a bunch of equations.
maybe it's a bunch of equations and different ones at different times. And so this distributed cognitive map maps out those relationships in different ways in different places. And they're all integrated to some extent, but not necessarily in any rigid way where you can say this is how it is from brain to brain, from person to person, or from rat to rat.
Or equations, at least I don't know, the equations I know is like they're good at modeling change of something that already exists. Like it's usually a varying change in a differential equation. So you already have to put the variable there. So you have to have already like specified something. But the brain, I mean, is that enough to understand the brain though? Because, the brain, do we want to, does it create stuff like a concept? So can you capture that how do you capture that with a system of equations? Because if the concept is not there to begin with, it's like something that emerged, then there's, you have to decide on a set of variables. So that's kind of like, that's kind of like the idea of, trying to understand maybe the basis of cognition in a way. And I don't know. So that's just.
Yeah. Well, I think
Yeah.
concepts do emerge. So when you have a bunch of paths that, Manhattan, streets and avenues crisscrossing, what emerges from that naturally are blocks. There's a thing called blocks.
Yeah.
And northwest corners of them. All these things.
We recognize those features, they make kind of a
Right.
pattern,
Right.
recurring, useful pattern that somehow becomes a concept.
So does that mean that there's no hope of finding a brain substrate of any concept like block? I mean, wouldn't we, if we're going to give up on that, maybe we should give up on it and like quit pouring all the money into it and stuff that we do?
I don't know that much money goes into it. But then,
Not enough.
to
Not
begin
enough.
with, no, most of the money goes into like very nitty gritty, computationally simple things. Sorry. I'm not bitter, I promise. But I think, when the concept, as it were, eventually finds its way into some kind of symbolic system, like a word, right? And we can find some neuron that seems to reflect only that in its activity, then all of a sudden we're sort of satisfied with that. So what was it, like the Jennifer Aniston cell in the hippocampus of humans, right? It doesn't fire to anything else. It's the Jennifer Aniston concept cell. And finally, we get
Been quiet for thousands of years of evolution.
So,
but then, got a lot of attention eventually. So I think as scientists, sometimes that's what we're kind of looking for, because if you say, oh, I say this parietal mapping of position in a route is evidence that the animal has this concept of route, which I stand by, right? But you're right. At any given moment, it's a different pattern of activity, right? So I have to say, oh, it's all these patterns of activity. All of these are consistent with this one concept. So now I have to think that I have to, in some sense, posit this notion that all these patterns of activity are capable of driving some neuron that represents that
I guess when I was
concept, and maybe that's a little.
I was thinking about something a little bit less profound than that. So that what we're used to with place cells is we think this cell's firing, so that means the animal is there. So if I really only knew about that cell and it was firing, of course the brain knows about all the cells, but if I just knew about that cell, I would know something fairly concrete about where the animal is.
Mm-hmm.
But less so with if the cell had a sequence of places it fires along a path. So in that case, I would have to look at more than one cell if I was the brain, because now this cell fires at this point, and this point, and this point, and this cell also fires at this point, but not at that point. And so by seeing that they're different, I could tell, you know, and of course, in a continuously varying thing like the result you get where cells speed up and slow down in a continuous way along the path, then you think about things like frequency components or something like that out of that whole thing.
Building block.
And so in that case, it's not that big of a difference, actually, to go from a system where individual cells tell you a lot, but you still have to look at them all because the animal's going to move around, or ones where you're always looking at all of them in order to tell the answer.
Right.
It's not that different. It's harder for us because as experimentalists we can't reconstruct what the whole network is doing from the data.
We're blind to it.
Yeah. But it's not harder if we imagine that we could know that. It wouldn't make the system harder to grok because it's not that different. So I don't know. I guess I'm trying to be a little bit more optimistic about how we could use our data to understand some of that stuff. And I still think that it's possible to address that Isabel's question about how distributed a concept could be, and we would still think the brain could use it as a concept. I mean, why couldn't a bunch of parts of the brain be contributing different parts? And I don't know, do we need an overall observer who's looking at all of that stuff to interpret, or could it just be embedded in all those places at once and be enough to control behavior and create our experience?
Yeah, I think it's embedded, and I think our appreciation of it, I think it's embedded, and I think our appreciation of it comes piecemeal, right? It's not necessarily that we have it all in our minds all at the same time. But I think something that can make it anyways a little bit less piecemeal is hierarchy. So as you move across the dorsal to ventral axis of the hippocampus, it's said that the place fields of neurons get larger, and that for grid cells, the space between nodes on the grid grows larger. I think that for place cells of the hippocampus, I think we need more data on that. That's really just from a couple of studies, which are fine enough. I don't mean to doubt them at all, but it hasn't been studied that well. But I think if you have this hierarchy, right, now you have neurons that are active. The idea is you have neurons that are active over the full space of the environment. They're still place specific. If your environment was 10 times bigger, you'd see that. But it's not. And so this neuron is active over the whole space. And on top of that, builds in all these other neurons that become active over smaller spaces within it. So that concept of a hierarchy is perhaps useful. And that's where Russell comes in again. Russell Epstein, I saw that work. Go ahead.
Actually,
talk about that.
About the tuning of the ventral cells, we did one of the studies that shows that. But what was interesting is that when you try to decode the position of the animal looking at the population of active cells, even though the fields are much broadly tuned when you move to the ventral pole, you get precise decoding in the ventral hippocampus, the same decoding that you get when you are recording from dorsal cells. So I thought they work like sensory systems, right? When you look at the sensory receptors, they are all broadly tuned, but our brain somehow can decode very precise information when you look at the population of active cells. So in that paper, we thought, why do we have this, redundancy in the spatial coding, some cells that are kind of have discrete firing or circumscribed firing, and then broadly tuned cells that only when they are all active together can provide precise information about the space.
Mm-hmm.
And we did some modeling in that paper to show that the brain is constantly trying to minimize interference, right? And so the dorsal hippocampus is very good at minimizing interference. But as you move to the ventral area may be much better to induce generalized responses that are also important when you are navigating in a space. And the brain has to have a balance between that process of interference and generalization, and they have to happen at the same time. When we are navigating in a space, we have to have a really clear idea of where we are in order to get to our goal. But if you happen to encounter some aspect of the environment that will lead to a dead end, and then that repeats in space, it would be wise if you learn to avoid taking those shortcuts in order to get faster to the goal. So it's both processes that have to happen at the same time in order to maximize the efficiency in which you move in an environment. So I don't know, we came up with that idea, and I have to keep exploring it further in relation to the ventral hippocampus. But I think that this differential coding has a purpose for navigation that allows us to deal with those problems. And perhaps because I have been focusing either recording from retrosplenium or recording from hippocampus, maybe if I were able to record from all these brain regions simultaneously, I would have a better answer of what's going on. I don't know.
Next time you'll have
Next
that.
time.
Next time. Someday we'll have the whole thing at our hands.
Yes. But it's intriguing.
That makes some sense. I usually, sometimes anyways, think of the cebiculum coming after CA1 as being the generalizer, where CA1 is more specific on the dorsal side. But maybe the ventral hippocampus should be thought of that way, not so much scale as generalizing. In some sense, I'm reminded of Sebastian Royer's data on ventral hippocampus on a switchback pattern that the animal was moving through. And the ventral CA1 cells had repeating fields at analogous locations along that pathway. But those fields themselves were hardly different than your standard dorsal CA1 place fields in terms of size. I
think
good.
it's very...
I stand corrected.
I find that it's
I like your explanation much better.
The ventral hippocampus is super intriguing to me, and I have been trying to push for this idea that is involved in generalization, but I don't rule out that it's about distance as well, because having, broadly tuned fields would be able to map larger environments much better, but I'm leaning towards the generalization idea because this is my bias. So
Plus, I think when it comes to discriminating versus generalizing, you come in as the experimenter and say, "Oh, I can take this population of ventral hippocampal cells and tell you precisely where the animal is." Right? Well, you've drawn some threshold for what constitutes the same or a different pattern. It's in the eye of the beholder. That's the eye of the experimenter.
Correct.
At that point. Whereas if you just drop your threshold for what constitutes the same versus different, you say, "Oh, well, that's the same firing pattern," right? And now you do just have a mapping over the same mapping over an entire space. It's all but that's really what we don't know a whole heck of a lot about. Right? We get this, and this is what bizarre.
It's
what happens
hard
to this signal in the brain, right?
Yeah.
Not what happens to this signal on my computer.
Yeah, no, of course.
Yeah.
But it's also very hard because when you go ventral, so if it's true like they act, say, as place cells, very large scale, now you need a very, 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 very large environment. in order to record. It becomes like it goes up with the square root 2, right? So basically, it just becomes impossible to experiment. So if you have a smaller environment, just say that that's a big place field, then it's either going to cover the entire environment, fires everywhere, or it's not going to cover anything and it doesn't fire anywhere, or maybe it fires half of the place, and then it becomes very difficult to say, is this a contextual signal, versus a spatial signal, because it might be a spatial signal that looks like a contextual signal because we're looking at a small environment, and now you get a place. Now you say place cells either fire or don't fire everywhere, versus, or it could be the other way around. It's contextual, and now we have a hard time kind of measuring the space. So it's hard experimentally. Like we're getting to a point in which, yes, you start seeing concretely, experimentally, this divergence between, okay, how the brain reads out things and what you need to do in order to read out these things and make them into a rate map or whatever, so that you can make sense of it. And at that point, you almost get to the point where it doesn't even, it's just, the question has to change. It can't be the same question, because otherwise we don't even have a way to think about it or answer it experimentally.
And I think, if I understand it right, that's what Bazaki was trying to emphasize a little bit in that latest book of his, the Inside Out.
Yeah.
Was.
Yeah, he elaborates a lot on that. Yeah.
Yeah, because neuroscience has this tendency to look at everything as an input and see how the activity maps onto the input, as opposed to saying, oh, okay, well, here's this activity.
Mm-hmm.
What happens down the line?
Right.
Which is a little harder and less fun, I guess.
Yeah. Well, yeah.
We all love our maps when we map out the locational firing of neurons, and you get these cool patterns and everything. It's very enticing.
They're so beautiful.
They are.
I still love when I see a good cell. It's so wonderful to see.
When you get the rate map over grid cells, something. So when he shows up, you run it, just get that hexagon. It's like, I don't know, something about it.
Yeah.
Okay, well, this has gone on. I think this is our winner for the official longest we've done in ages. I have the heart to stop you guys. This is great. Thank you for joining us, Doug Nitz. This has been Neuroscientists Talk Shop. Thanks, guys.
Bye.
Bye.